Alright, so in this video I'm going to talk a little bit about and outline the process of DNA replication. As you'll see, it's a relatively simple process. First, we know that DNA is a winded up double helix. So there's one nucleotide on each side that bonds with each other, and that double helix needs to open, and so helicase will open up the double helix, and SSBs, or single-stranded DNA binding proteins, are going to stabilize each end of the double helix that have been opened so that they don't bond with each other once again, so that each end can stay open. And in each end, it either goes from 3' to 5' or 5' to 3'. And when DNA is replicated or synthesized, that synthesis action has to go from 5' to 3'. So it's going to go, if the top strand is from 3' to 5', it needs to go the opposite way, so it's going to go 5' to 3', and that top strand that goes 3' to 5' is called the leading strand, because as you'll see, DNA polymerase is what adds those bases, is going to be able to just go straight through in line with the replication fork. And once that replication fork is opened up by the helicase, tension starts to build within the rest of the DNA double helix. And so topoisomerase is going to make little cuts to relieve that twisting pressure so that it doesn't explode and the whole system is broken. So going back to that leading strand, before DNA polymerase can begin synthesis on the strand, a primer needs to be put down so that DNA polymerase can do that. And DNA primase makes an RNA primer, which acts as the primer for DNA polymerase, which is a little bit confusing. The DNA primase makes the RNA primer, which helps the DNA polymerase, but that's how it goes. And so that's pretty, outside of that one little part that's confusing, that's pretty simple. That's how it works with the leading strand. As that replication fork opens, the DNA polymerase will make nucleotides to match, going this way, kind of opposite towards, going 5' to 3', opposite of the 3' to 5'. But on the bottom strand, which is called the lagging strand, it's not quite as simple. Because the lagging strand goes 5' to 3', the DNA polymerase has to go in fragments away from the replication fork. And because it has to be going away from the replication fork, like I said, it makes fragments, and these fragments are called Okazaki fragments. And these fragments are eventually bound together by DNA ligase, which ligates the fragments and combines them all together. And once those processes both finish in the leading strand and the lagging strand, you then have two strands of exact copies of DNA, and that's how DNA replication works. Like I said, pretty simple process. There's maybe one or two things that are a little confusing, but outside of that, it's pretty simple. And the way that I would relate this process to the gospel is that within this process of DNA replication, there's lots of little problems and solutions that come up. And there's not one enzyme or one protein that fixes all of the problems. There's little solutions to each problem. And DNA replication is a super accurate process, unlike other processes in our body. And for example, when the DNA needs to open, there's an enzyme that opens it. In order for the base pairs to not pair again, or the bases to not pair again, there's a protein that comes and fixes that. There's not, like I said, one protein or one enzyme that fixes all of the problems or causes everything to work. There's each and little individual parts that cause the whole of DNA replication to work, and work very well. And I liken that to the gospel in that each of us is a unique individual with unique talents and abilities. Obviously, we can develop talents and abilities, and we're supposed to, but we each, as members of the church and members of God's kingdom, bring something different to the table that God needs for us to do. There are certain situations maybe that we're better equipped to help with. Maybe some of us are better at teaching, some of us are better at leading, some of us are better ministering individually to people rather than leading a whole congregation. And people with all of those different skills and abilities are needed to do God's work in the church, and I don't necessarily say solve problems within the church, but to help people and to build the kingdom of God as He would. And that's how I would liken DNA replication. Each little part has its own part that it does, and because of all of those little parts doing what they're supposed to do, the whole process goes well. And this is a process that goes on in our bodies, in all of our cells, 24/7. And so that's how I would liken that to the gospel. But yeah, that should be everything.